Thank you very much for your interest in our study, Affinity Matters for IgE blocking activity of allergen-specific antibodies. My name is Maria Strobel, and on behalf of all co-authors, I'd like to show you what we found. It is well known that allergen immunotherapy induces IgE-blocking IgG antibodies. Those IgG antibodies bind allergen, and then allergen does not bind IgE on the surface of effector cells, such as basophils or mast cells, which prevents their degranulation and reduces allergy symptoms. This concept was further confirmed recently by passive immunotherapy trials with allergen-specific monoclonal antibodies. This was shown in the context of cat dander and birch pollen allergy. In this study now, we were interested to find out whether IgG antibody affinity has an influence on their IgE blocking capacity. And in order to find this out, we generated patient-derived allergen-specific monoclonal antibodies. So, of an individual who had undergone successful immunotherapy and who had an induction of allergen-specific IgG1 antibodies that were IgE blocking, we took the PBMCs and isolated RNA. This RNA material was the basis for performing Fab yeast display, by which we generated human allergen-specific monoclonal IgG1 antibodies that we screened for IgE blocking capacity. We further took two of them, K1 and K2, and improved their affinity, also using Fab yeast display, and by this generated 3 descendants of K1 and 2 descendants of K2 that were shown to have a 2-fold to 5-fold improved affinity. We wanted to test whether those affinity improved antibodies are now better IgE blocking antibodies by basophil activation test. And in fact, in basophil activation tests, the affinity improved antibodies were more potent at inhibiting allergen-induced basophil activation. So yes, higher affinity of IgG results in a stronger IgE blocking activity. Thank you very much for listening, and if you're interested in further details, please feel free to go on and read our publication. Thank you very much.